called Specificity of optical connection.
Thanks. So I'm not going to talk about any behavior. All the experiments that I'm going to talk about are done in a slice of brain in a dish. So it doesn't do much. But we hope that what we learned by looking at cortical circuits has something to do with the kind of things that Richard talked about and Bill talked about. Part of the brain that all the data you're going to see today comes from is the rat visual cortex, so it certainly has something to do with visual processing, but we also think that the basic organization of cortical circuits is likely to be similar across most, if not all, cortical areas. And the very basic things that I'm going to tell you about today, I think, are likely to be true across at least primary sensory areas and probably across most of the cortex. As we've learned more and more about cortical circuits over the years, it's become clear that at every level of organization that we look at, we see that circuits are really precise. Things are very selectively connected to each other. So early on, people looked at cortical connections, and you can see that certain cortical areas were only connected to certain others. And then within the cortex, you see different layers, and you see that certain layers in cortex are only connected to each other, or columns, functional columns, and they're specific. Well, it turns out that what I'm going to show you today is that two neurons that are right next to each other and look exactly the same or are the same type can have very different connections and are selectively connected. And but whether or not those connections are specific depends on the type of connection. So I'm going to start out by telling you that there are different types of connections, and there's specificity of these connections, but the way that these specificities relate to each other depends on the type of connection. So we'll go over this in some detail. But before I get to that, I want to mention a couple of general ideas that have been around for a while about how connections might be organized. And both of these two very basic ideas have been consistent with the kind of data that have been available. One is that when people started to make paired recordings between two different neurons to look at whether they're connected to each other, you can see that connections in the cortex are sparse. That you might find that only 1 out of 4 or 1 out of 5 neurons that are even right next to each other are actually connected to one another. So this could give rise to the idea that connections in the cortex are sparse, and that sparse randomly connected networks are what are doing the computations in the cortex. A very different idea comes from the kind of experiments or models that John Hopfield started making in the 80s, where he applied learning rules to connections and said, well, maybe with sort of Hebbian rules, you can create specificity of connections. And that according to these rules, they may create groups of neurons that are selectively connected to one another. So I'm going to show you data that says that certain kinds of connections, feed-forward and recurrent connections, define populations of neurons that are specifically interconnected to each other. But the feedback connections and inhibitory connections don't follow those same rules and have different specificity that are not they're not specific. They don't respect themselves as defined by these feed-forward and recurrent connections. So to start with, we're looking at this in brain slices, and I'm telling you they're feed-forward and recurrent connections. I better tell you what I mean by that. So in any cortical area we look at, if you look at the anatomy of the connections and also relate a lot of this to function, I don't have time to go in the background and end up in as much detail as you might like. But we can think of the sensory input coming into a visual area, it goes to the middle layer, layer 4, so that's a feed-forward connection. Layer 4 then connects to layer 3. That's a feed-forward connection. Today, I'm going to be talking mostly with respect to neurons in layer 3. So we're going to define connections for layer 4 to layer 3 as feed-forward connections. These then project out to connections. In addition, within layer 3, there are recurrent connections. The cells are connected to each other. Layer 3 connects to layer 5, and those layer 5 cells make feedback connections back to layer 3 cells. So I'm going to call that feedback. 4 to 3 is feed-forward. With respect to layer 3 cells, 4 to 3 is feed-forward. Within layer 3 is recurrent. 5 to 3 is feedback. And then there are also many different types of inhibitory neurons. And I'm going to, I can't distinguish between them with the data I'm going to show you today, but I'm going to talk about inhibitory connections as well. Now, there's lots of data now that say that connections can be highly specific, particularly for different cell types. So in any given part of the cortex, you might find different cell types and find that they get input from different sources. There's also some data suggesting that even individual neurons of the same type could have specific connections. And I'm just going to show you one example of that is part of what motivated the study that I'm going to tell you about today. And that's that people reported from pairs of neurons, say in layer 4 and layer 3, they see that they're connected to each other only about 20% of the time. Now, if this were a sparsely or a randomly connected network, and if you plotted the distribution of the number of connections between cells, if you had 0 connections 80% of the time, you'd expect that the next most likely number of connections would be 1, and following up to 2 and 3 and so on. But in fact, what is seen in this study by Feldmeyer from Stockman's lab is that in 13 cell pairs that were connected where they actually identified the size of the synaptic contacts, that in every case there were either 4 or 5 synapses. So that suggests that these connections are probably precise. There's probably something about the neuron this cell is targeting that it liked a lot. So how does that kind of specificity relate to specificities of other types of connections? The feed-forward connection is specific. Is that grouping of neurons that might be created respected by recurrent connections, is it respected by feedback connections, and what it would give you. So the way that we've addressed these questions is to take advantage of a method called photostimulation that I'm going to tell you about. And what we did is to record simultaneously from 2 pyramidal neurons in layer 2-3, and use this photostimulation, which untages glutamate. I'll show you a few slides about that. And it causes a group of neurons to fire action potentials, but relatively asynchronously. So if there's one neuron that fires an action potential and connects to the 2 cells you're recording from simultaneously, then because this connection is highly reliable, 95% reliability, I can't show you that data, but it's 95% reliable, but if it's connected to one cell and the other cell, there's a high probability you're going to see exactly synchronous synaptic currents in both cells simultaneously. But if it's 2 different cells that we're providing input, you expect that to not be synchronous, and we can calculate what the chance level of synchrony is, as well as that that comes from the shared common input. I'm going to just toggle through these slides because that way I won't stand in front of you. With photostimulation, what we do is we bathe a brain slice in caged glutamate. This is what caged glutamate looks like. And then we focus light from the UV laser to various locations in the brain slice. This causes the glutamate to become uncaged. That depolarizes neurons that have cell bodies close to the stimulation site so that they'll fire action potentials. And it's important to know that the only neurons that fire action potentials when we do this are ones that have cell bodies very close. And in all the experiments I'm going to show you today, we've eliminated stimulation sites that are near the laminar borders where there's uncertainty about what location the cell might have been in. Or the cells. And then we can record synaptic currents, in this case in one cell, that follow stimulation. And you infer that if you see evoked currents, they must come from the cell you stimulate, from a cell that was stimulated. In the experiments I'm going to show you, we record from 2 cells simultaneously. Before I show you that data, I want to show you the timing of action potential to show you the idea that the groups of neurons that are firing action potentials are firing relatively asynchronously. To show you data where we actually reported action potentials simultaneously in 2 cells to show that, in fact, during trials when you stimulate and record simultaneously from the 2 cells, the synchrony of action potentials is not any greater than you just expect from it. If you shuffle the trials. So there's actual connections between those cells aren't creating more synchrony of action potentials than just you get from the distribution below. So you can see that when you photostimulate, here's from many neurons in layers 4 and layer 5, that there's initially a fairly high probability you'll get action potentials, and it falls off. And if we record action potentials simultaneously in 2 neurons, you do a cross correlation in the matched windows, use you see this hump in the middle that says that because the stimulus tends to cause active images could require in this distribution in relation to the stimulus that there is some probability that action potentials would be synchronous. But when we look at the cross correlation in the shifted between shifted windows, there's no difference, and essentially this is noise. So we calculate a number called the correlation probability, which is the probability that if an event occurs in one cell, that you'll also see a synchronous event in the other cell. So we can use these numbers also to look at synaptic currents. And the first data I'm going to show you is to show you that the feed-forward connections and recurrent excitatory connections define populations of specifically interconnected cells. So here's data from a typical pair of pyramidal neurons. This is cell number 163,292, 293 right next to it. And this is typical data, what we see from most cells we report from. If we stimulated this site number 1 and record synchronous simultaneously synaptic currents through two cells, you can see that they come roughly at the same time, but not real well synchronized. We see the same thing for trial number 2, a lot of synaptic currents. And when you look at the cross correlations, you see that there's really essentially no difference between the matched trials and the shifted trials. We'll come back to layer 5 later. You can see there's actually something different. Well, it turns out these are those 80% of the times when the two layer 2-3 cells are not connected to each other. But when the layer 2-3 cells are connected to each other, we see something different. So now these are connected cell pairs. So about 20% of the time, when we report from two layer 2-3 cells that are right next to each other, they are connected to each other. They have recurrent connections to each other. And now you see that these correlation probabilities are much higher in the matched trials than in the shifted trials. And about 30% of the time here, when there's a synaptic current that's detected in one cell, then there's a synchronous synaptic current in another cell above the values you'd expect from the chance correlation of spikes. So these cells share about 30% of the cells that connect to one cell also connect to the other cell. So this is what we see with the, when there are connected cell pairs. And that's true both for layer 4 and layer 5. So now if we look at the distributions from all the cell pairs we reported from, and then separate them into cell pairs that are not connected to each other versus cell pairs that are connected to each other. And then we look at the stimulation sites in layer 2-3 and layer 4, come back to layer 5 later. We see there's a very clear difference between the connected cell pairs and the unconnected cell pairs. When the cell pairs are connected, their correlation probabilities are high, meaning they share a lot of common input within layer 2-3. They also share common input from layer 4. So these connected cell pairs share common feed-forward input from layer 4, and the recurrent connections maintain that selectivity. When they're not connected, they don't receive as much common input. So the feed-forward and recurrent excitatory connections define populations of specifically interconnected cell groups. Well, the feedback connections and the inhibitory connections turn out to not respect those same cell groups. And you already saw this data. When we look at layer 5 input, we see that the correlation probabilities are high or moderate, but there's no difference between connected and unconnected cell groups. So there's some kind of selectivity here, we think, but this input from layer 5 isn't respecting the same groupings of cells that are defined by the specificity of the feed-forward and recurrent. And we see the same kind of thing when we look at inhibitory connections. We can measure inhibitory connections using the same method, except now when we record from the cells, we change the holding potential of our recordings. And now these outward currents currents are inhibitory currents. And now you can see that regardless of whether we're simulating layer 2, 3 or layer 4, and regardless of whether we have connected cell pairs or unconnected cell pairs, we see very high correlation probabilities. So these cells tend to share common inhibitory input regardless of whether or not they're connected. So again, the inhibitory connections don't respect the same improvements. And here for the populations of cell pairs, you see essentially no difference between connected and unconnected cell pair. Correlation probabilities are high, both for layer 4 and layer 3. Layer 5 isn't included here because there's not a lot of input from layer 5. Inhibitory input from layer 5 to layer 3. Now, I don't want to leave you with the idea that all inhibitory connections are non-specific. These recordings are biased toward very large synaptic currents that are fast rising, probably on the cell body. I think the inhibitory connections are very important because there's a lot of specificity in inhibitory connections. It's just that in these experiments, we happen to record the synaptic currents that are most easily detectable and reliable. So fast spiking basket cells, when we do paired recordings between them and layer 2, 3 cells, we see that they have very reliable connections and have strong fast rising inhibitory currents. But when we record from other inhibitory cell type pairs, those are much less reliable and small and hard to detect synaptic currents. So we don't think those are coming into play here in these recordings because, as you can see, these are very large, easy to detect fast rising currents. So this schematic diagram summarizes what I told you then. With respect to layer 2, 3 pyramidal cells, layer 4, feed-forward input from layer 4 defines populations of cells that it's selectively connected to. So one set of layer 4 cells connects to one set of layer 2, 3 pyramidal cells and a different set of layer 4 cells to another one. And that cell grouping is reinforced by the recurrent connections within layer 2, 3. But feedback connections from layer 5 don't respect those same cell groupings, and inhibitory connections also don't respect those same cell groupings. And that's all. Thanks. I think it's very likely that they would have similar receptive field properties, right? And in the rat visual cortex, which we looked at, rats, for example, don't have orientation problems. But they do have orientation selectivity. So one possibility here is that these cells don't have the same orientations. I would expect that if we looked at an animal like a cat, which does have orientation problems, we still would see probably the same result. Alice and paired recordings in cats see the same kind of thing, that looks sparse with respect to 20% of the cells that be connected. And a while ago, Greg DeAngelis made recordings in cat visual cortex where he recorded simultaneously two neurons that are right next to each other. You will see that they can have different receptor field properties. So in the cat, there's columnar organization, but there are also things in any animal, any cortical area, that are also not organized. And neighbors have different specifics. That's another question.
Yes, we have time for another question. Otherwise, we can make a couple of announcements. So the lunch will be on that side. We will guide you towards it. For the poster sessions, we will set them up in the corridors that are here. And so we can help you with the